In this video, we will discuss about this how the mKRAS trigger inflammation or immune suppressions. So some of the things we have discussed in our previous videos. So here we will discuss those things and some of the new things. So first we will discuss this mutated KRAS, active form and the inactive form. So from the inactive form to active form, it goes from this KRAS GDP when it interact with this GTP. When it interact with this GTP through this GEF, it will form this KRAS active form that has this KRAS GTP. So it will GEF will take away GDP and insert GTP. And GAP, what it does, it will remove one of the phosphate, and then it will again form this GTP to GDP, and that is KRAS inactive form. So however, in the case of mutated KRAS, what will happen is that this GAP hydrolysis weakens. So KRAS remains continuously in the active form, and this active form of KRAS will triggers changes within these cells that will cause the inflammations. So some of the main changes are shown in this figure. So we will start with this signaling pathways, that it will upregulate MEK ERK signaling pathways. It will leads to the upregulation of this gene FOSL2, and it will cause the release of cytokines such as CXCL28. And this CXCL28 thereby helps in this regulatory T cells formations, which plays a role towards immunosuppressions, which will inhibit the activated cells, immune cells. Other things that it will upregulate is IL-6 receptors and can interact with this IL-6, and thereby the phosphorylation of this STAT3 will be improved. We have discussed the role of STAT3, how they help in this polarization of the macrophage. So this phosphorylation of this STAT3 will trigger the M2 macrophage polarizations from M1, and they also helps in this regulatory T cells formations. Both of their role is to support tumor growth and suppress immune response against the tumor cells. And this because of the change in the signaling pathways or change in the homeostasis in the pancreatic cancer cells, it will have more reactive oxygen species, and it will more reactive oxygen species is there, then it will trigger the survival pathways, that is AKT pathways through this ERK, MAPK, and this survival pathways, since it is triggered continuously, it will cause the cell proliferations or tumor cells proliferations. And the other things in terms of the cytokines, it will release IL-1 beta, and this IL-1 beta can cause upregulations of the CXCL1 PDCD1, and that will helps in this trafficking MDSC to the site of the tumors. The MDSC roles is to suppress the immune response. The interleukin 1 beta has another role that will also upregulate CXCL13, and this CXCL13 upregulations will cause high expressions of this PDL1 on the B cells, and this PDL1 is the immune suppressions ligand that can interact with this PD1 receptors on the immune cells, and thereby inhibit the immune response against these B cells. And the cytokines, this IL-1 alpha, it can trigger this NF-kappa beta pathway, and this NF-kappa beta pathway has cause more pro-inflammatory response, and the complex formation of this TBK IKKe will also enhance, and this will cause this more M2 type macrophages polarizations, and this M2 type macrophages will result in this tumorigenesis or tumor proliferations. And the one thing that it inhibits is this HMGA2, and this HMGA2 will result in the downregulations of this CXCL10 and 11, and downregulation of this will finally results in the CD8 plus downregulation of the CD8 plus T cells, or these are the main cytotoxic T cells that release the lactic granules to kill the tumor cells. But in the case of mKRAS mutations, this it will trigger in a way that it will leads to this downregulations of the CD8 plus T cells. So all these things will either helps in this tumor suppressions or no, not tumor suppression, immune suppressions or tumor proliferations. So if you need to understand more about this topic in detail, can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.